right. Hello, everyone. Welcome to this afternoon session. It's my pleasure to introduce our first speaker, Marinka Zitnick. Marinka is currently an assistant professor in the Department of Bioinformatics at Harvard Medical School. Prior to joining the Harvard faculty, she did her postdoctoral research at the Computer Science Department at Stanford. Marinka holds a bachelor's degree in computer science and mathematics and a PhD in computer science, all from the University of Ljubljana in Slovenia. And her research focuses on the foundations of AI for scientific discovery and biomedicine. Over the years, she has been recognized by many best paper and research awards. Most recently, she's has been recognized as a 2023 Kavli Fellow from the US National Academy of Sciences. Today, Marinka will talk about multimodal learning on graphs. Marinka?
Okay, hello everyone. I hope you can hear me well. Very excited to be here with you today, and thank you for this kind introduction. The title of my talk is Multimodal Learning on Graphs in Therapeutic Science and Medicine. So I will be covering some of the methodological advances in this area, as well as how those advances enable new opportunities for applications in drug discovery, development, and personalized medicine. So we have seen in recent years advanced computational techniques, in particularly and deep learning models being increasingly incorporated into scientific and medical discovery across all stages of research. And that includes using the models and to collect better data sets and process and augment data sets that are being collected for scientific and medical research, using the models to generate medical and scientific hypotheses, and then finally also use AI models in combination with some simulation and large data science methods in order to really enable new kind of discoveries that might not be possible using traditional scientific methods alone. There are several, well, apologies for that. There are various stories and examples of success that include the ability of AI to be able to model and predict structure of large molecules, such as proteins, from their pure amino acid sequence, the ability to scatter and parse through large chemical libraries in order to identify what are small number of compounds and molecular structures that might have certain desirable properties for research in material discovery and drug development, as well as for modeling new therapeutic candidates in order to identify whether a particular molecular and therapeutic candidates might actually bound to molecules in human body in such a way that they would exert positive therapeutic effects. So we have seen these advances and in proof of concepts of how AI models can really accelerate and augment scientific discovery in medical research. And throughout this talk, I will highlight two key enablers of these advances. And the first is that of graphs. So many data sets in biological and medical research have underlying geometric properties, geometric structure, or they might explicitly come in the form of relational or structure-based readouts. And that is true for data generated across biological scales, from the level of interactions between proteins and genes within individual cells to the level of interactions between cells within a specific cell type or tissue, and all the way to interactions between diseases and phenotypes within an organism, such as human body, and then at the level of ecosystems or populations where it is interesting and relevant and often necessary to be considering relational structural nature of the data that we are looking at. And the second key enabler is that of being able to look at multiple data types or multiple data modalities simultaneously when modeling a particular biological or medical phenomenon. Specifically in biology and medicine, there is rarely a single data type that captures the entire complexity of a biological phenomenon that we're studying, and it often requires really generating data from the level of genes all the way to phenotypes in order to more holistically understand a particular complex problem that we are investigating. And so with these two big opportunities in mind, I will highlight throughout this talk and focus on two key topics. So the first is that of leveraging multimodal data in the form of developing methods that can learn explicitly on highly heterogeneous, highly multimodal relational structures that we represent as knowledge graphs. And I will motivate the development of the models in this area with problems that arise in drug discovery development and therapeutic science broadly. In the second part of the talk, we will discuss contextual AI models, which are a new kind of AI models that can adjust their outputs to biological context in which they operate in order to generate predictions that are more actionable and more readily translate to downstream experimental investigation. So let's start with the first part, which is that of knowledge graph-based models. And so we know already that data in biology are diverse, they are multimodal and span across biological scales. The last 2 decades have generated just massive amounts of transcriptomics data, information on structures of large molecules and how they physically interact, information about how those molecules cooperate and work together in groups known as molecular pathways in order to perform certain biological functions, and also how really these molecular interactions manifest there then in the level of human patients and human populations in terms of diseases and then treatments that are being developed from them. So we have these very different types of data, and so what we are interested in developing and where the focus of state-of-the-art AI and machine learning research is to find a way to represent this massively heterogeneous data in a consistent manner and then develop large models that are trained in self-supervised manner without explicit labels that can then be easily adapted through a process known for fine-tuning to solve a number of downstream tasks and make predictions for the tasks in a way that does not rely on massive amounts of labeled data. In extreme case, that would be zero-shot learning without reliance on any labeled information. And so that's the class of models that operationalize this paradigm of training and pre-training large models in a self-supervised manner and then fine-tuning them to a broad array of tasks are known now as general-purpose, generalist, or foundation models. And so in this talk, we will be discussing one model in this, of that type that we refer to as TxGNN, and that stands for Therapeutics Graph Neural Network. And that's a graph neural network approach, which is a large pre-trained model that is trained explicitly on graph structure data. And the graph structure data that we'll be looking at is a large knowledge graph that brings different types of biological data and represents it in a unified manner. So what we have on the left is the input to TxGNN, which is a comprehensive knowledge graph that spans over 17,000 clinically recognized diseases. And for each and every of those diseases, we have rich molecular information available in the sense of what are specific genes and gene products that are associated with the onset of that disease and how that disease phenotypically manifests itself in human patients. And so TxGNN will be trained on this large knowledge graph with the goal that once trained, it can process various therapeutic tasks. And by therapeutic task here, for the purpose of today's discussion, we're interested in tasks known as indication or contraindication prediction. So at the very high level, you can think of these problems as tasks where we want to find whether a particular small molecule drug, which we'll refer to as therapeutic candidate, might likely treat a disease, and we'll refer to that as indication, and or alternatively, whether a particular therapeutic candidate and drug should not be prescribed and given to human patients because it might actually make disease worse, and this is known as contraindication. And so what we want to have a single model that will be able to predict indications and contraindications across the entire span of 17,000 diseases. So the way we will do that, on the first critical component for building foundation models, is to have a massive data set that is represented in a consistent, unified manner. And so the way we think of large data sets and how we can represent it in a unified manner is through knowledge graphs, which are these heterogeneous graph structures where we have nodes that represent biological and medical concepts of different types. Here they are denoted by different circular structures in this plot shown on the left part of the slide. And these nodes that represent distinct biological and medical concepts are related to each other through edges or links that carry specific semantic meaning. And so when we adopt this kind of knowledge graph perspective, we are able to then consider a broad range of biological entities, ranging from proteins and genes that are that encode the proteins, as well as how those proteins are relate to diseases based on a known understanding of biology, what are the functions and biological processes those proteins participate in through gene phenotype and gene pathway relationships, and so on. And so a particularly appealing a property of knowledge graphs is that they present this massive data set on which we can then define mathematical pre-training objective in a consistent manner, and then bring very different, and allowing us to train a single model on varying diverse data types. So for TxGNN model in particular, there is one rationale that we are operationalizing with the model. And that rationale can be thought of in the following manner. If we are interested in designing models that would reliably and accurately predict therapeutic opportunities for a specific disease, the way we intuitively think of that is that for a disease of interest, which is shown here on the top of the slide, there is some understanding of what alterations in the underlying genes and proteins in other biomolecules are associated with the onset of the disease, right? So we know what are molecules and interactions that when dysregulated, perhaps through mutations or some other alterations in human body, might increase the risk of the developing of disease being developed in a patient. And so what we want to do, if we want to find a therapeutic candidate that would treat a disease, the question is, can we find a drug that would actually counter the effects of those disease alterations and counter the effects of molecules and interactions that are being perturbed in an unwanted way in a disease? So really, the effective drug would be the one that if we take a dysregulated molecular interaction network, we would want to mitigate or counter the effects of alterations in those molecules. And so that means that in order for us to build a model, we need to have information about what are these dysregulated processes, molecules, and interactions that lead to the disease. And for a specific therapeutic candidate, what are molecules that that candidate binds to and affects them and exerts its therapeutic effect? And if we can match these two aspects, the molecular mechanism of a disease and the therapeutic mechanism of a drug in a productive manner, we expect that we can extract patterns that will then be associated with indications, positive treatment, or contraindications, negative treatment of the therapeutic candidate. And that can effectively allow us to then prioritize therapeutic candidates for treatment. And so that is what TxGNN model does. TxGNN model, as I mentioned, is a knowledge graph-based graph neural network that is trained on prime KG knowledge graph. And what it takes as input, a candidate or query disease and a query therapeutic candidate shown on the left. And through a process and a series of neural message passing operations that we'll see in the next few slides, it produces as output scores that represents the likelihood that the input therapeutic candidate will likely be indicated for the disease, and the likelihood that the therapeutic candidate would be contraindicated for the disease. And in addition to making predictions, what the TxGNN model can perform, it can extract evidence from the knowledge graph that can be used, that it relies on when making a specific prediction. and that evidence can be then visualized in the form of mechanistic paths and associations that the model has relied on when making prediction, and presented to domain experts who can use that information in order to decide whether a particular prediction made by the model is to be trusted and sufficiently reliable for downstream investigation. If you go one level down to a more like technical view, what is behind that TxGNN box, it's really a large knowledge graph message passing algorithm, so that when a queried disease and queried drug are provided as input by the user or by the developer during training time, we find what are the nodes in the knowledge graph that represent the queried drug and disease. And then the graph neural network explores local neighborhoods in the knowledge graph of the drug and of the disease in such a way that it will learn what are the patterns of molecular mechanism of the disease and the patterns of the therapeutic action of the drug. And if those patterns are such that are activated towards indication, it will make prediction that the query drug might likely be indicated for the disease. The use of knowledge message passing algorithms into graph neural networks is really not new, but the critical enabler of TxGNN model is a module that we refer to as disease pooling. So disease pooling is an idea that allows TxGNN to find in the knowledge graph what might be other diseases that are similar in their mechanism of molecular mechanisms to the query disease, and then borrow information from those other diseases and transfer that information to the disease that we are currently looking at. And this is crucially important to be able to make accurate predictions for those diseases that are very poorly understood and those diseases that have few or no existing treatments, because that effectively allow the model to transfer information from other diseases that might be distant in the knowledge graph to the query disease, but are better understood from the standpoint of biology. So slightly more detailed view of TxGNN is particularly the disease pooling module is shown on the top of this slide, where we see that the knowledge graph message passing really operates on the local neighborhood of the drug and disease, here shown as these large blue and yellow circles. And then once the local neighborhood is investigated and neural messages are propagated along edges of the local neighborhoods of the drug and disease nodes and aggregated in the form of drug and disease level embeddings, the disease pooling module actually scores similarity between the query disease and a number of other diseases that appear as nodes in the knowledge graph, and finds what the most relevant diseases and then bring them over to enhance the query disease level embedding. And that allows us to get a much richer representation for those diseases that otherwise have very sparse or incomplete local neighborhoods, meaning that a current understanding of those diseases from the scientific and medical standpoint would be very poor, and we can kind of supplement that information from other diseases. So once we train TxGNN model, we can use it to make predictions across this entire range of human diseases. And so I want to highlight specifically the evaluation setup that is realistic of real-world use, but is rarely employed in modern machine learning research. So existing work and research today would generally evaluate the model and use it in the following manner. We would assume that we already have some number for a specific disease, shown here on the left as this kind of circular shape, that there are already some available treatments for that disease that would be represented as edges between disease node and the FDA-approved drug nodes representing approved indications for the disease. And that the disease has well understood mechanism. And with that assumptions, making predictions around what are additional drugs that might treat that disease are relatively easy to make, because there are already examples, known examples of available treatments for the disease. And so this is a relatively easy task that can be solved through supervised learning. However, what we really want to solve here is this problem of zero-shot therapeutic use prediction. We have which is shown on the right part of the slide. We have a disease. However, that disease node in the knowledge graph is not linked to any of the drug nodes, meaning there is no existing approved treatment for that disease. We want to be the first to predict what candidate treatment for that disease would be. And that's really, really challenging, and where existing models fail. You might ask, well, but yes, great question. Yes. Absolutely. So great questions. I will repeat the question for the online audience. So the question was around these two distinct setups. And while the right one might be more interesting and relevant for real-world use, one would expect that a model that can solve the right-hand problem should also solve the left-hand problem. And the answer, yes, that's absolutely true. And I will show results for both of these setups in the next few slides. What I want to briefly touch upon is why is this zero-shot setup so interesting and relevant from the scientific and medical standpoint. And there are numerous reasons why this is the case, and I will briefly mention two key challenges that can be addressed with the zero-shot learning. So first is that there are actually many human diseases for which there are no existing treatments. In fact, there are over 7,000 rare diseases which are defined as diseases in the US that affect less than 200,000 patients per year. But collectively, though these diseases are so plentiful that they collectively affect a large number of people in the US and worldwide. Unfortunately, only 5% of rare diseases have any FDA-approved drugs. And even for diseases with some approved treatments, finding novel drugs that would treat them could offer alternative options that might cause fewer or less severe side effects, or replace drugs that are ineffective for patient subpopulations. So there is a real need for this kind of model capability. And the other is that if one would find that an opportunity to repurpose the use of existing drug for a new disease, that can lead to much faster translation of that drug to the clinic and lower development cost. Because the development of a new drug completely from scratch takes on average between 12 to 15 years and can cost $2 to $3 billion. But if one is able to repurpose an existing drug for a new disease, that the process of developing that repurposing strategy is compressed and also much cheaper. And however, the problem is that most repurposed drugs to date are results of accidental, non-systematic findings. So this is a clear argument for why computational and machine learning models in particular would be really helpful for such approach. So if we now really are convinced that zero-shot setup can be relevant for us, let's see how well TxGNN can perform on both the harder but more relevant zero-shot setup, as well as more traditional machine learning view of predicting associations or links between drug and disease nodes in the knowledge graph, which is where most of existing work lies. And what I'm showing here on the right plot is the aggregate is the performance of TxGNN model and a state-of-the-art graph neural network, and I will show additional results on the next slide. But these are the summary slides that show performance of TxGNN and state-of-the-art GNN model that is not TxGNN under the standard evaluation setup, which is this easy prediction model. And we can see that the improvement offered by TxGNN, measured as area under precision recall curve, is relatively modest, and we are not surprised by that. However, what we are really more excited are these two blocks of bars on the right part of this plot that show performance of TxGNN when it is asked to make predictions for diseases that it has never seen during and encountered during training time, and none of the treatments for those diseases during training time. And there we can see improvement that is by TxGNN that goes close to 49% and 46% for diseases of cell proliferation. We've done numerous more detailed analysis of the model that I won't have the time to go to, but we effectively kind of systematically benchmark the model on this zero-shot setup, where we designed the test set that the model was evaluated on after training in such a way that all diseases in the test set were diseases with known approved treatments, diseases that were forming distinct disease areas that were not encountered by the model during training time, as well as diseases that have limited molecular understanding, meaning that their local neighborhoods in the knowledge graph are relatively sparse. And so what I'm showing here on the bottom of this slide, we can see 8 plots where on the y-axis of every plot is area under precision recall curve, so higher value indicates better performance. And then we have on the x-axis different machine learning models. So in addition to TxGNN, which is shown on the far right of every plot, there are various others, and graph neural network-based models, large language-based models, network science models that are based on network proximity, as well as more standard optimization techniques that TxGNN is compared against. And we can see that across different therapeutic tasks for both indication prediction, contraindication prediction, and across different disease areas, TxGNN really gives superior performance. And that was very exciting for us to see. It was so exciting that we asked ourselves, well, if the model can make correct predictions in this benchmarking setup, can it make novel predictions that we would be also interested in looking at? Yes. Yeah, so the question was, what was the actual task here? So the task here is that of indication and contraindication prediction. So given a drug and its known mechanism of action, so knowing information about what are the drug targets and the molecules the drug binds to, and how those binding of the drug to those targets changes the molecular behavior of the cell and then the organism, can we predict whether that specific drug will be indicated, so likely have positive therapeutic effect for the disease? And so this is done without consideration of patient-level data that I showed on the previous slide. But what I'm showing now on this slide are then patient-level analysis, where we, after performing extensive benchmarking of the model that we briefly looked at on the previous slide, we're interested in looking at some of the novel predictions that the model would make, meaning that those are predictions about indications that associate an existing drug with a disease that currently do not correspond to an FDA-approved drug. And our reasoning was the following. If these novel predictions that the model makes have any value, then what we would expect is that those novel predictions would be enriched in off-label prescriptions that clinicians are currently making in large healthcare systems. And so to test that hypothesis, we teamed up with our collaborators from Mount Sinai in New York, and we processed an EHR system of 1.2 million patients from New York that covered 480 diseases and 1,200 drugs. There is really a massive amount of work behind what is just a single or less than one slide. Those who worked with this kind of healthcare data might appreciate the amount of mapping and data processing and sample size calculation that was required to do that. But after the EHR data set was processed, we had knowledge about the signature of a patient population and how often the specific patient population is prescribed a particular drug in an off-label manner, meaning it's not approved by FDA, but the clinicians are applying their medical expertise and have the ability to prescribe the drug to disease in certain circumstances. And what we would expect is that our novel predictions should be highly enriched, found much more often in those off-label prescriptions than what we would expect by chance. And that's what is shown on the right part of this slide in the form of these 4 blue bar plots, where we can see that the red horizontal curve shows the enrichment of known FDA already approved drugs, and we would expect that enrichment should be the highest for existing FDA approved drugs. However, if you look at for every disease top 1 prediction that TxGNN made, that enrichment of that prediction in off-label prescription in EHR system is the highest, and it goes quite relatively close in resemblance the level of prescriptions that we are seeing for real FDA approvals. And in fact is near 1 time larger than if one would look at those predictions that were ranked at the top, at the bottom of the list that was predicted by TxGNN. And so we were very excited to see that. We did another study where we used a technique known as temporal data set split, where we retrained TxGNN model on all the data that was available up to June 2021. And then we asked the model to make novel predictions. And then we waited for some time and looked at whether those novel predictions, whether any of novel predictions made by the model were actually recently approved by FDA between June of 2021 and today. And so what the table here is showing, it's showing 10 examples of such recent FDA approvals and also predicted ranks for those approvals that were made by TxGNN before any of those approvals were really made. In another study, we leveraged TxGNN model to identify therapeutic opportunities for an emerging pathogen. And those predictions made by the model were then later verified through laboratory experiments, providing an order of magnitude better hit rate than what would be possible if this experimental screening of predictions would not be guided by AI. Of course, then the natural next question then arises, which is, well, if we really trust this model so well, shouldn't we transition them in some meaningful way to real-world use or real-world implementation and deployment? And what is really a bottleneck of doing so, and what prevents us from doing so? And is this really just a trivial problem? And it's really not a trivial problem, because developing a machine learning model alone is not sufficient. One needs to think about actually developing tools that allow domain experts, which would be clinical researchers, drug designers, and developers, to interact with AI models, ask queries in an interactive manner, get predictions, understand what are the data the model relied on when making predictions, and do that in a continually improving manner. And so one of the that was very exciting for us, and so we embarked on this journey of designing an interface for the AI model that is clinician and scientist-centered. And to evaluate that interface and the quality of explanations that for predictions that TxGNN model made, we evaluated predictions and explanations across a spectrum of usability metrics in collaboration with human experts, where we wanted to do a user study showing predictions made by the TxGNN model, and then ask a number of experts in the field what is there to see if there is scientific consensus on utility of those predictions, if predictions made by the model together with explanations that the model produced allow them to more quickly reason whether a prediction that was made is trustworthy, is of any relevance and quality to be followed up, that they would follow up on it or not. And so in our usability study, we considered, and were specifically interested in this question of how to present and extract explanations from this large knowledge graph. We want to extract really small, compact, sparse amount of part of the knowledge graph that can be visually presented to the user and provided as evidence of the data the model relied on when prediction was made. And so the way we generate those explanations is through a counterfactual algorithm known as GNN Explainer, which is a model that was developed in my group a few years ago. But here we were really interested in ways how to visualize those explanations in a way that that human experts would kind of be able to quickly reason with them. And so we can fundamentally think about 3 different types of explanations that we can extract and present to the user. Every explanation should really provide some evidence of what is the knowledge of the drug that the model relies on, and what is the knowledge of the disease that the model relied on when making a prediction. And so the simplest way of looking at explanations is just to look at neighboring nodes of the query drug and query disease and visualize those nodes that have the highest attention scores by the model. And that's option A. And option B is different, which is that of what we will visualize are really these subgraph structures. So not only direct neighbors around query drug and query disease, but they're local neighborhoods or structures. So we need to have subgraph level visualization for that, which was option B. And option C is different than A and B in the sense that it assumes that there are certain reasoning paths known in ML field as meta paths, which are a sequence of biological entities that start with the query drug and then go to the query disease. And so our usability study showed that these path-based explanations perform significantly better than node-based or neighbor or subgraph-based explanations across all 3 usability metrics in terms of scientific consensus, confidence, and the amount of time a domain expert needed in order to make sense of predictions made by the model. And that is true across a variety of diseases and variety of drug candidates that we tested in this study. And also results of this analysis have informed how could we really practically create a tool or an interface within which TxGNN model can be integrated. And so this model is currently live. It exists and lives on txgnn.org website. If you go to that link, you will see a version of the model that allows you to enter a query drug and query disease and see predictions made by the model, as well as explore different ways of visualizing the model. And we are very excited to have had a chance to really explore and apply now this model to a broad range of diseases through our collaboration network here at Harvard and beyond. There is another concept that I would like to bring, an opportunity that I would like to bring up, which is that of in the research development involving large language models, we have this amazing opportunity to start to reimagine how humans can collaborate or communicate with AI models. And so that involves not only communicating and interacting with AI models through CSV tables or Excel spreadsheets, which is what we traditionally most often have done, or even through these kind of more intelligent interfaces such as TxGNN interface that I just showed to you. But actually, one can communicate would potentially communicate with AI models in a very flexible manner in the form of plain text interfaces, in the form of prompts that would allow you to kind of flexibly describe any disease or phenotype without requiring that disease to be codified a priori in some medical vocabulary. And so that brings us to this notion of that in addition to knowledge graphs being in some way multimodal, but in the sense that all the different modalities they can bring together have explicit relational structures, there are other data modalities that are of great interest to us and of great relevance. And of course, that brings us to the question of how can one model and incorporate text-based information in the form of plain text, English or in some other language. And what's even more exciting from the standpoint of biology is that of modeling biological sequences of amino acids and nucleotides that represents the sequences of genes and those proteins that are encoded by genes. And it's a reasonable expectation and tremendous amount of evidence in scientific research that integrating this sequence level information into TxGNN model could further improve the model performance, as well as integrating then text-based natural language could further improve and enhance ways for human experts to interact with these models. And so one of the first questions that we were asking in this regard is really how can large language models that were primarily and are primarily developed for natural language, how applicable they are to biological sequences, or actually language or sequences that we encounter in biology, which you can also think of language in some way, but it's completely different with its own set of vocabulary, grammar, and semantic that is driven by evolutionary relationships and kind of fundamental biological processes. And so one question that we were very interested in understanding is how do existing large language models and their pre-training objectives, if at all, impose any relational structure on the way they generate embeddings and representations of words and tokens? Is there any inherent geometry that those models are learning on its own? And by reviewing over 70 different popular large language model objectives, we have found and provided a theoretical analysis showing that most NLP-derived, so pre-training methods that are derived from natural language processing field, really do not impose explicit or deep constraints on these latent embedding spaces of words. But those explicit constraints are really crucially needed in biology, where we have explicit relations and constraints that are given to us in the form of biomolecular interactions, such as, for example, interactions between proteins in the form of protein interaction network. So that motivated us to develop a framework that we refer to structure-inducing pre-training framework that can combine objectives for standard large language modeling and various NLP-derived objectives with objectives that allow us to leverage graphs or other structural information in an explicit manner that is available at pre-training time. And the results are incredibly encouraging. What I'm showing here on the right part of the slide is one experiment where we were interested in evaluating whether we can predict biological functions of proteins more accurately by integrating both their sequence-based large language model with a structure-based model that considers protein interactions between proteins encoded by those genes. And so what we can see here are these different curves show performance of different models, where the blue curve shows performance of structure-induced pre-training framework that combines both structure and sequence-based models, which is considerably better than using language modeling on biological sequences alone, shown in green, or using then regular NLP-derived masked language modeling, shown in yellow. And also, all these different pre-training strategies are better than using kind of more standard established supervised machine learning model that does not rely on any pre-training at all, which is shown in red. There are numerous other studies and benchmarks that we have developed for this field, for this problem that I would be happy to discuss in offline and also follow up on that, and the details are described in the paper. One specific, very exciting project that I want to mention here that is enabled by this work is that all of a sudden we are not a priori limited anymore to make predictions for those diseases that are a priori characterized and exist in formal medical ontologies or medical vocabularies. And so that might seem not particularly exciting or significant to you, but what that means is that with these text-based large language models combined with biological language models and knowledge graph models, then human experts can use text-based models and describe the phenotype or the disease of interest in plain text in the form of a paragraph that is then used as prompt to the model, and that model can then produce predictions without that phenotype or disease necessarily really existing as part of current formal medical vocabulary. And so this ability of doing predictions with unbound vocabulary is something that I think is crucially important for problems that are being studied at frontier of science and frontier of biology, which are often those problems that do not yet exist in formal medical vocabularies and terminologies. Okay, an important question, and apologies for this not being shown the way it should be. An important question to ask at this stage is whether it would, how can community actually leverage and use some of the models and data sets that we are developing, as well as other groups who work in this exciting field at the interface between core foundational AI and, on the other end, drug discovery, development, and therapeutic science. And so as part of that, my group has founded an initiative called Therapeutic Data Commons, which is available at TD Commons AI. And so TD Commons AI serves as a meeting point between, on one end, biomedical scientists who really know what are the bottlenecks of and key problems and unsolved tasks in modern drug discovery and development, and on the other end, machine learning AI scientists who can develop robust, well-performing algorithms once those problems are mathematically defined. So Therapeutic Data Commons is an initiative with the goal of, whose overarching goal is to access and evaluate AI capability across stages of drug discovery and development, from drug design all the way to drug repurposing, and do that across therapeutic modalities. We are very excited to have a number of collaborators that are all listed on the website. And we currently have over 200,000 active users of AI for a broad range of diseases and drugs around the world. And in the Commons, you can really find many models already implemented, benchmarks, standardized performance and evaluation metrics, as well as ready-to-use data sets for developing new models or training large models on existing biomedical knowledge. In the remaining time that I have, I want to briefly mention this other, I think very exciting direction that is made possible by recent advances in AI, which we refer to as contextual learning. And what I mean by that intuitively is the idea that we would really like to have AI models that could adjust their outputs automatically based on biological context in which they operate. So what's the motivation for that? So let's forget about biology for the moment. So let's look at the word apple. So apple is a polysemic word. You can grow an apple, you can buy an apple. And so the difference between the meaning of the word really is resolved by the context within which that word appears. In the context of this word, the context is given by the sentence or the paragraph within which the word apple appears. And it might appear within the context that indicate we refer to the apple as a fruit, or we might refer and use apple in the context of apple being a company. So this notion exists and is prevalent in biology as well. For example, we can look at H2AFX, which is a gene that is pleiotropic gene. And so that means that the function that this gene plays in human body depends and varies with respect to where that gene acts. And so you can think of the function and the role of that gene is also resolved via biological context, which in this situation is now not a sentence, but is actually biological context given in the form of tissue, or even more precisely given in the form of cell type within which that gene is activated and expressed. So with that understanding, and with this example, the question becomes, is this just a contrived example, or is this really, really strongly prevalent and important problem? And if so, why do we need contextual models? And the answer to that is contextual models are crucially needed because genes and proteins can have distinct roles and play distinct functions that vary with biological context. While nearly every cell contains the same genome, the expression of those genes and the functions of products that those genes encode can vary considerably across cells, across tissues in human body. And the problem, however, is that this is not recognized by existing machine learning models that are all producing these representations or embeddings of genes, proteins that are context-free. When you get for a single protein, a single embedding vector that represents the information about that protein in one specific context, or just integrated across all those contexts. So can we do better? And so the model we developed that is a contextual graph neural network model is called Pinnacle, and was really spearheaded by a fantastic PhD student in the lab, Michelle Lee. And her idea was that we can leverage large molecular cell atlases that were generated in single-cell genomics over the last decade in order to provide biological context information in the form of cell types, within which then we can train graph neural networks to make predictions about the functions and roles that proteins play. And so this project had two big components. One was that of putting together data from structural biology in the form of protein networks and data from single-cell genomics, which was a different field of biology that has generated large molecular cell atlases. And the other big component is that of developing such a contextualized model that we refer to as Pinnacle. And so the way Pinnacle works intuitively, it is being optimized through a self-supervised objective that is being applied on a large multi-scale network. So what I mean by that is the following. At a very high level, you can think that the input to training Pinnacle model is a large number of protein networks. In this example, I'm showing in this specific study that I will mention, we considered 156 cell type contexts. And for every cell type context, we built a protein interaction network. And so the goal and objective of Pinnacle was then to take this set of protein networks and learn representations or embeddings for every protein that exists in any of those 156 cell type networks in such a way that proteins that interact within a specific cell type context would have embeddings in that cell type context that would be attracted to each other in the latent embedding space. And if proteins do not interact in a specific cell type context, they would be repelled from each other in that latent embedding space. And so this can be done through a type of self-supervised learning known as contrastive learning, where this latent embedding space can be learned. And it's learned in such a way that we get a massive latent embedding space where for every protein we have not anymore a single embedding, but we have a set of embeddings for every cell type context. And so this is in sharp contrast with what existing models can provide, which is that of providing a single context-free representation, whereas what Pinnacle can provide is a set of these protein representations for contexts in which the underlying genes are activated. And we were interested in knowing whether this additional flexibility that contextualized representations provide can help us to tackle some of the challenging problems. And I will, in the interest of time, just briefly mention one of these three applications, which is that of contextualized prediction and what I mean by that. So far, in the first part of the talk, and I would expect also some of the earlier speakers today and yesterday might have mentioned this notion of, or the task of predicting drug-target interactions, which can be thought of as tasks where one is interested in knowing whether a specific drug or compound currently in the development will bind to the protein that it is expected to target in a specific way. And one would want to do that because that protein might be associated with the disease that we want to treat. So it's a really crucial canonical problem in early drug development. And so what we were interested in asking is it possible to advance this task beyond how it is currently being studied in the literature by incorporating information about where are those target proteins active in what cell types and in what tissues. And so that is what we refer to as contextualized prediction. We first train a Pinnacle on a set of 156 cell type specific protein networks. And then in the fine-tuning stage, we define a contextualized prediction as a downstream task. And the question was, is a given protein a strong candidate for a specific therapeutic area and the drugs in that therapeutic area? As additional insight, we're interested in knowing what cell type is most predictive of that protein being candidate target for a specific drug. We focused on two disease areas for now, which are those of rheumatoid arthritis and inflammatory bowel disease. There is a reason for that, because those are two diseases that are relatively well represented in existing single-cell molecular atlases. And there is some known knowledge that supports the fact that therapeutic targets of drugs for rheumatoid arthritis and inflammatory bowel diseases really require, and their efficacy depends on the cell type in which those therapeutic targets are expressed. And so what we have done then is applied Pinnacle in this pre-training, fine-tuning fashion to a set of drugs for RA and separately to a set of drugs for IBD, and for each of drug in these two therapeutic areas, ask Pinnacle to predict therapeutic protein targets for each drug in these two areas, and then measure performance of Pinnacle relative to state-of-the-art context-free representation learning models. And we're excited to see that Pinnacle representations can predict drug targets of existing RA and IBD therapies significantly better than context-free methods, indicating that incorporating contextual information can really help improve model performance. And even more excitingly, is the results shown on the right that shows that one can take state-of-the-art representations that are learned provided for proteins based on structural information, and effectively enhance those representations by concatenating representations of those models with Pinnacle representations that bring in this contextualized information. And in doing so, we were able to predict protein interactions involving key proteins that are being studied in immuno-oncology and are relevant for immunotherapy. And this suggests that Pinnacle representations can be used also as drop-in replacement or enhancement of existing structure-based representations of proteins, which is a very active and vibrant field in our community. For those who are doing working on follow-up experimental studies, leveraging some of our novel predictions, we were excited to provide not only predictive scores and raw predictions made by the model, but also pinpoint what are these specific cell type, contexts, and types of cells that the model has identified as most predictive of targets for these two disease areas. And we are currently working with our collaborators from Brigham and Women's Hospital and Mass General on using Pinnacle on an ongoing clinical trial for a novel therapy that's being developed for rheumatoid arthritis. So let me wrap up my presentation for today. In this talk, we broadly touched upon two big topics. The first topic is that of using and developing knowledge graph-based models in order to provide novel predictions across a large number of diseases and a large number of existing therapeutic candidates, and do so in a unified formulation without relying massive amounts of labeled data that traditional supervised learning methods would require. In the second part of the talk, we discussed the idea of contextualizing machine learning models in a way that they produce outputs that are adjusted to specific biological contexts within which those models operate. So all that would not be possible without just a wonderful group of students and fellows that I work with. And I would also encourage those who are interested in learning more about our research or just trying out some of the models or using the data sets that we are processing and using to reach out to me or check out our lab website or the Therapeutic Data Commons Initiative or our AI for Science initiative to learn more about this exciting field. And I'm also very happy to take questions if there are any in the remaining time that I have. Thank you very much.